Dear students, now I am going to start the practical class. This is a virtual practical class. So hope we will be offline soon. In this class, I am going to introduce you to the practical class, practical zoology. Then today I am going to explain you regarding microscopy, the first practical class. So, what are the importance of zoology practical class? You know already zoology is the subject which deals with the study of animals, their anatomy, physiology, embryology, genetics, evolution, classification, habits, behavior and distribution. This is a science and practical oriented science. Appropriate experiments and investigations are the key for higher learning, clarification and consolidation of theory. What all you have studied in the theory, you can clarify in the practical class. So, it is the part and parcel of a science subject. So, without practicals, the study of science is not going to complete. Practical activities are motivational and fun, enable you to apply your knowledge and to understand the zoology. So, it is the practical aspects in zoology is very inspiring and it is very fun giving aspects because you get thrilled looking into the animals in the preserved form or else in the live form or else to do some behavioral experiments these animals, all these are really it is giving lot of fun. So that's why, and you will apply your the theoretical knowledge to understand the animals in a proper way. Practical activities can stimulate interest in the subject and helps in better learning and memory retention. What all you study the characteristics and the anatomy and all this, if you in the practical class you are going to see all those and you will have a better learning and experience, and as well as it is helping in the long term memory retention. You will remember for longer duration. It is in the theory, you will forget quickly. So that's why it is practical is very important. Practical works inspire you and may stimulate a lifelong interest in animal science. This is what you will see. The practical activities in zoology may stimulate you to develop a hobby. So, for example, you may start collecting information regarding identifying different types of butterflies or birds or worms. So, all this it's like it will start from the zoology practical class. So, this you may continue in a biology career or else you may take up some other career after your BSc, but this animal interest, what it is evoked here in the practical class, it will be lifelong experience sometimes or lifetime interest it will develop in you. So, high quality appropriate practical work is the central to effective learning in science. It is remember, it is just not theory is important, it is in science is practical is important. It is practical work make you it is the better learning, effective learning experience you will get from the practical work. Practical in science stimulates your creativity, curiosity and critical thinking. That's what I said. So, these practical work stimulate you to study the insects. Those are present in your backyard or else in your garden. You to study the spiders in your home or lizards in your home. So, it's like it evokes lot of curiosity, and also you start questioning and you start observing the things more carefully and methodically. This is what the effect of practical zoology. Practical in science stimulates your creativity. Of course, you will become a creative person by the involving yourself in practical work, and that may stimulate your critical thinking, and you may end up with end up with the taking up a research career. So it promotes you engage with the scientific method and encourages active learning and problem solving. So scientific methods and it is supporting active learning, not a passive learning. So you just inquire and you study by yourself. So this is what you can see, you motivated towards learning and problem solving. So it allows collaborative working, it's something science projects and all, you have to work along with your classmates. It is a collaborative working, and it provides opportunity to the it's develop a soft skill in you to adjust with the other people, and also it is applying these the practical data and processing it in the with the help of a statistical method or mathematical method or computational method, that additionally you will be equipped with a lot of skills. So this is the advantage of practical class. So in what are the learning objectives in this first BSc first semester zoology practical, the paper 1, you are studying diversity and functional anatomy of non-chordates. So here the learning objective is to understand the non-chordates with respect to taxonomic position, general characteristics, organization, phylogeny, from the phylum Protozoa, it starts with phylum Protozoa to hemichordata, the last one you are going to study. So in this evolutionary sequence, you will see you are going to study the taxonomic position, general characteristics, organization, anatomy and phylogeny of phylum Protozoa, Porifera, Coelenterata, Platyhelminthes, Nemathelminthes, Annelida, Arthropoda, Mollusca, Echinodermata and Hemichordata specimens in the practicals. What are the learning outcomes? So you should be able to identify and classify an invertebrate based on its morphology and functional anatomy. So this is you will develop here in the practical class, and you should be able to recognize the uniqueness of an animal. So every animal is different, and its role, ecological role, you should understand. So all this you can do, you have time to study individually these animals in practical class, and you should be able to appreciate the diversity of invertebrates and to be sensitive towards its conservation. So all these are the learning outcome of practical paper 1. So today I am going to give you the introduction to some the common microscopes you use in the zoology practicals or in the zoology lab. So in that way, the first class I am going to deal with the microscopy. So then microscopic slides, you are also studying to the so many the protozoans they are all in slides, and anatomy you will study in slides. There will be microscopic slides study, there will be specimen study, some animals, the bigger one, for example sea anemone or octopus, all these are in bottled specimens. You are going to study these bottled specimens. Then you are going to mount some the appendages of prawn, or else the some protozoan culture on the slide and stain it and observe it. So that is the part of a practical activity, that is called mounting. Then the demonstration. So we may demonstrate some the systems through the by in models or some the presentation. So that is you have to practice those demonstration. These are all the part of the practical paper 1. Today I am going to discuss about microscopy. Microscopy is a technique of using microscopes to view samples and objects that cannot be seen with the unaided eye. So, with in your the eye capacity is limited, so the objects that are not within the resolution range of the normal eye, you have, so for example, if you want to view the amoeba or paramecium, you need microscope. So, or else if you want to take out a particular salivary glands from a cockroach and want to study in detail, then you need a dissection microscope. So, these microscopes are part and parcel of biology practicals. Optical microscopes are referred to as the light microscope, is a type of microscope that uses visible light and a system of lenses to magnify images of small objects. So, these three basic type you are going to study here, or you are using constantly in our lab: simple microscope or dissection microscope, compound microscope or light microscope, and stereosomes or stereo microscope. So, this is a simple microscope, or else it is called a dissection microscope. You might have seen this already. So, it is just like a lens, it's a how you use a lens to view the small object, the same thing you can see. A simple microscope uses a single lens, this is the lens, to magnify an object. When a tiny object is placed on the in this platform, you are placing a slide or an object, and it is you can view it. The focal length of this convex lens will be smaller than the focal length of the lens. Greater will be the magnifying power. So, this is the, you have studied in your physics classes, in your lower in PU classes. So, this is the lens, it is the biconvex lens. So, when you place an object, it enlarges almost 10 times. It is the power here, it is the resolution power is 10x, you can see 10 times it enlarges. So, this microscope is having all this, there is a metallic stand and it support all these parts. Then there is a stage where you keep the slide. Then there is a mirror to focus the light on the object. When you place a the object here, and you can focus light. Then there is a biconvex lens. This is in this, you will see biconvex lens. So, there will be adjustment knob. This is adjustment knob. So, what are the application of this microscope? The maximum magnification of this simple microscope is about 10x, which means that the object will appear 10 times larger by using this simple microscope. And it is used to study the algae, fungi, and that's what whatever you are mounting, the appendages of prawn, or else salivary glands of cockroach, or spiracle of cockroach, mouth parts of cockroach, all these you can study under this, or else you can mount with you by viewing under this. That's why it's called dissection microscope. And this is a compound microscope. You have seen already in your PU classes and in high schools, and you are using this constantly here. And this compound light microscope uses visible light for illuminating the object, and contains combination of lenses to magnify the object. This is not a one lens here, it is a combination of lenses, objective lenses, ocular lenses. So, objective lenses will be here. So, it's a rotating nose piece. This is called nose piece. You can rotate, and you can, so it is a 10x or it is 40x, 100x. So, it's different magnifying lenses will be fitted here, and you whatever depending on, for example, if you want to see a paramecium, you may use the 10x. If you want to see the detail, the nucleus structure of the paramecium, you have to use 40x, like that. You can see depending on the structure. You can use different lenses here. That's the advantage. Ocular is the eyepiece. Then there is a condenser. You will find there is a condenser, and which focuses the light on the object. So there is a body tube. This is called body tube. So these what are the mechanical part? It is the one is the base, then there is a C-shape arm you will find, then there is a stage where you keep the slide, then light passes through the specimen, and this is there is a lens which focuses the light on the specimen. Then there are adjustment knob, coarse knob, and fine adjustment. So you are adjusting depending on the objective, the size. So this adjustment knob, coarse adjustment and fine adjustment knob. So what is the principle here? Image formation. This also you have studied in your physics classes. Light from the specimen is projected by the objective. It spreads evenly across the entire image plane at the diaphragm of the eyepiece. The objective lens at first formed a real and inverted. This is the objective lens. It focuses and it magnifies. It may be 10 times, 40 times, 100 times. Further, it further magnified by the eyepiece for 10 times or 5 times, you can see. So that is the advantage. Is a combination of lens and the magnification is far higher than that of simple microscope. So it is the common microscope we use. You are using for 3 years in our lab for viewing so many things like protozoa, then the sections of different worms, appendages of prawn, then to view the the larvae of echinoderms, larvae of some other the forms, then the scales of fishes, sections of liver. Likewise, it's handy. Even the mitosis, meiosis, chromosome study, all this you will able to do with this microscope. You can do a lot of things. You are constantly using this. You should know how to use this. Then the last one is the stereo microscope. This is it is not, you are not using constantly, so it is only helpful in viewing a living object or a object and dissecting it, or you are doing taking out something out of this, then this will be very helpful because it gives a three-dimensional view of the specimen. That's the advantage. This is also called stereozoom. So it is having two optical paths. There will be two lenses in this is the objective and these are the eyepiece. There are so they are of different focal length, so that you get a 3D image. This is what is the stereo zoom. So this is ocular lens, and this is a diopter setting. That means focusing differences in the left and right eye. So the objective lens, the stereo microscope, have two separate objectives. So two separate objects you will be able to find. Then the focus knob, as usual, it focus. Then there is the light source you will find from this. There will be usually electrical bulb is fitted in this box and helpful, and you are placing the object here. And there will be a clip to hold the slide, or so this is very helpful in the dissecting the Drosophila larva, to take out salivary glands and salivary gland chromosomes. So likewise, something small, dissecting out cockroach salivary glands and all this. This stereozoom is very useful. So this is about microscopy. Thank you.